Well, thank you, and good morning. Can I have the first slide? And is there a remote? So I'm going to talk about how the vaccines were developed so quickly, and then move into, at the end, how disappointed I was that they were not deployed well enough. So I'm currently at the Morehouse School of Medicine. I used to be at the Vaccine Research Center at NIAID at NIH in Bethesda, where I was deputy director of the Vaccine Research Center. But now I am at Morehouse. These are my disclosures. I'm an inventor on vaccines and monoclonal antibodies for several viruses, and I consult for a number of pharmaceutical companies. But right now, I'm mostly focused on my work at Morehouse School of Medicine, which is celebrating its 50th year. And this is a list of the presidents, former presidents of Morehouse. The bottom president, Dr. Valerie Montgomery Rice, is our current president. And the school is very focused on equity. It's equity and health justice are in its mission statement. It's one of the reasons I'm at Morehouse as I leave the laboratory. So we're faced with an ongoing burden of emerging and re-emerging virus diseases. Our public health, our traditional public health approaches are adequate and effective, but limited in their scope. Fortunately, we have a whole new set of technologies over these last 15 years that have changed the balance and have allowed us to make vaccines and conceive of vaccines in a very different way, using these new approaches. And historically, as vaccines have come, it's largely been because of new technologies. So the first two vaccines listed were before we knew about immunology, before we knew what a virus was, and those were made by Jenner and Pasteur for smallpox and rabies, really in animal tissue, in the spinal cords of rabbits and dogs or on the hides of calves. But as we progressed into an era where we could grow viruses in eggs, it allowed us to make the influenza and yellow fever vaccine. When we, Enders and all, learned how to do cell culture and grow viruses to higher titer, it allowed us to make live attenuated viruses or whole inactivated viruses. Then we moved into the era of molecular biology, where we could grow now high-density bioreactor cells, high levels of virus, and also make recombinant proteins and virus-like particles, reassorted viruses, et cetera. Now we're in a completely new era, I think, that is typified by the SARS and RSV programs, where a lot of the work will depend on structural biology. So we can now have atomic level structures of our vaccine antigens and design them with much more precision. Why do we focus on vaccines? Because vaccines have an amazing track record of effectiveness and safety. So this is just data from the United States, where I'm showing the peak year of cases for each of those viral diseases and how it's changed now in the modern era, going from measles, 500,000 cases a year, down to 59, and we no longer have wild polio or smallpox or congenital rubella in the United States because of our vaccine programs. WHO recently published this review of the expanded program on immunization over its 50-year history, showing that we've averted over 150 million deaths and added 9 billion life years to the planet, largely from the measles vaccine. The biggest contribution, 2/3 of that, is from measles vaccine alone. So vaccines work, and they've worked. What has worked so far mostly has come from the 20th century technology. And now we have 21st century technology with all those different types of things that we can do that have changed the nature of vaccine development from empirical antigen selection to precision engineering. We can understand things now at an atomic level. We've gone from cell culture to chemical synthesis, from individual products to platform technologies, making it possible now to develop vaccines in months or years instead of decades. At the Vaccine Research Center, I went there in 2000 when it was first founded, and these are some of the outbreaks that we responded to with vaccine development programs. I'm not going to go through that entire list, but I want to call your attention to Zika, because in 2016, a lot of things started changing for how we prepared for future pandemics. For one thing, we started focusing on families of viruses, and I'll explain that in a minute, but we thought that we could get ahead of these pandemic threats in a more proactive way by finding generalizable solutions using these new technologies, especially structure-based antigen design, and develop a plan around prototype pathogens representing the 26 virus families that can infect humans. And we also started thinking about speed, and there's new ways of delivering vaccines, and using platform manufacturing, you can shorten the regulatory process and develop better ways and faster ways of making vaccine products. So a lot of thinking was started from that time of Zika. So the idea of a prototype, you know, there's lots of viruses on Earth. I'm showing you, you know, there's over 11,000 species of viruses on Earth. But if you only look at the viruses that infect humans, you can break them down into 26 families and about 150 species. And we like to divide them by entry mechanisms. So I'm referring to the class 1, 2, and 3 fusion proteins that get viruses into cell, or non-envelope viruses. And we think that if you organize the viruses in that way and assign pathogen-specific investigators to understanding the viruses in those 4 boxes, and then back them up with development tools like we had at the Vaccine Research Center, where you could manufacture products and bring a research project all the way through to a clinical project, that we could make about 30 prototype vaccines for 30 prototype pathogens and develop interventions through phase 1, and for the other 120 or so viruses, at least develop concepts through animal studies, so that we would be ahead. And this idea has been adopted now by the WHO, by CEPI, the Coalition for Epidemic Preparedness Innovation, and by NIAID, especially through their revamped grant program that is generating the data we need on structure and replication pathways and entry mechanisms and things like that that you would need to know ahead of time to make a particular intervention. So I'm going to tell you briefly this story of 3 viruses. Respiratory syncytial virus on the left is a filamentous virus. The SARS-CoV-2 on the right is a spherical virus. You see the knobs on the small particles next to the cilia. Those are the spike proteins, making it the obvious target for a vaccine intervention. And then Zika, which is a flavivirus. It's in the middle, and I'm not showing it to scale. It's actually about half the size of a coronavirus, so it's only about 50 nanometers in diameter. But these 3 viruses together created the story that allowed rapid COVID vaccine development to occur. First, with RSV, which is a virus I've studied my whole career, starting with trying to understand the vaccine-enhanced disease that occurred in the 1960s. So starting with 20 years of work on safety, on vaccine safety, we eventually came to the point where we could capture the structure of this protein shown on the right, the F protein, that is responsible for virus entry. It's the protein that helps the virus get into cells to start replication. And we were able to identify antibodies shown on the left and then on the top of that protein that could hold the protein together in a particular shape so we could achieve the crystal structure of that, what we call pre-fusion form of the F protein. These proteins undergo a big rearrangement to pull the membranes together for the virus and cell to help the virus get in. And so keeping the protein in this pre-fusion form is the original native functional form of the protein. And it turns out that those antibodies that could hold it together from the top were about 100 to 1,000 times more potent for neutralization than the only antibody that had been used in humans, palivizumab, that was used to prevent severe RSV in newborn infants and premature infants. And that was done in about 2012. And over the last 10 years, that work has now resulted in 3 different vaccines that are now approved for use in humans. Protein with adjuvant, protein alone, or mRNA delivery of a protein are all approved for the elderly. The protein only is approved for maternal immunization, and there's an antibody, the one that I showed in the picture, that was modified to be used for prophylaxis in young infants. So we now have RSV vaccines or prevention approaches for both the 0 to 6-month-old child or the elderly. So that was a 10-year process, but it was about a 70-year process from the time the virus was discovered. The other story from the flavivirus is that in 2016, when Zika emerged, we used what we had learned from West Nile a decade earlier to make a DNA vaccine, a nucleic acid type vaccine. And it was a reasonably effective vaccine in animal models, but by the time we got it into phase 2B efficacy testing in South and Central America, the epidemic was over and we didn't get an answer from that vaccine. But at the same time, as part of a consortium of US government agencies, we also were helping other groups test their vaccines in our non-human primate animal model. And we tested 3 mRNA vaccines during that year of 2016. One from University of Pennsylvania, Drew Weissman's academic group, one from GSK, and one from Moderna. And all those studies were done in 2016. Some of them were published rather late, but this was when we learned that mRNA was now a better nucleic acid vaccine than DNA. And that wasn't true 2 years earlier. In 2014, it wasn't working that well, but now in 2016, it was working. So the other story is about coronavirus, and because of the RSV work, we wanted to extend that observation to other envelope viruses, and so we started working on coronaviruses. We were unsuccessful with MERS, coronavirus, and SARS-1, and so finally landing on an endemic coronavirus, HKU1, we were able to solve the atomic level structure of the spike protein shown there on the left. Jason McClellan was the postdoc in Peter Kwong's lab who I'd done all this work with, and then Andrew Ward on the left used, instead of crystallography now, cryo-electron microscopy to solve this structure. And once we had this structure, we were able to find ways of stabilizing that protein, in this case with 2 proline substitutions at that place where the protein pivots to turn up, and it largely stabilized. But in addition to preserving the epitopes at the apex of the protein, it also increased expression of protein from transduced cells, which makes a big difference when you're delivering vaccine through gene-based delivery. And so when this outbreak was reported at the end of 2019 out of Wuhan, and when we learned on January 6th of 2020 that it might be a beta coronavirus, because of the last 6 years of work we had done on coronavirus biology and structure and mechanisms of neutralization, we were in a place where we sort of knew what to do, because we had been working with Moderna for the last 3 years, showing that a stabilized coronavirus spike protein for MERS could be delivered as either mRNA or protein and protect against lethal challenge. A lot of that work was done by Kizzmekia Corbett, who's shown there on the left, along with Jason, who did the structure. And this work was all informed by the prior work on RSV, the thinking about pandemic preparedness, the findings with our nucleic acid vaccines, and that mRNA was now working well, and the importance of platform technologies to make things go faster. And even some of our work on the top from our work on RSV vaccine safety became relevant for this new virus response. And so when the sequences came out on January 10th, we were able to quickly make the reagents that we needed to make protein, to solve structures, to make assays, immunize mice and animals. And then within about 65 days, we were able to start a phase 1 trial, and within about 6 months, start a phase 3. And as everyone knows now, the authorization for use in humans came within 11 months, less than a year. So within a year, we had a vaccine that was very effective and ready for use in humans. And it was also effective, found to be effective after distribution. So the effectiveness studies, post-marketing studies, showed that there may have been as many as 20 million lives saved during just 2021. The problem, and this is something I wrote about on the 3rd anniversary of the pandemic in 2023, is that despite all these new technologies, despite everything that we can now do, and we can do almost everything we need for solving most of our problems, but the problem that became so evident during the pandemic is that it wasn't benefiting everyone. Our policies and practices weren't keeping up with the technology, and we were not we were having a problem with both uptake and with access. So if you look at the mortality curves day by day during that early part of the pandemic, and then the vaccine distribution, which is plotted on the red line, you can see that once the vaccine got started, you could start gradually showing a decrease in the overall mortality. But sometimes people ask the question, why is there so much gray mortality left when all those vaccines had been given? And the answer is that after July of 2021, as you went into the Delta wave and then the other different variants that came along, most of the people who were dying from coronavirus were unvaccinated. These were people who decided, this is US data, these are people who decided not to take the vaccine even though it was available to them. So since the vaccine has become available in most people could have been vaccinated by July of 2021. Most people that have died from coronavirus have been unvaccinated. And most of the benefit came during the Delta wave in the fall of 2021. So most of the deaths averted happened in November of 2021. They've continued to accrue, but a lot of this benefit of the vaccine happened in that early Delta wave. And if you look at who was protected, most of the deaths averted happened in high-income countries, some in middle-income countries, but very, very few in low-income countries. And that was not because of uptake, but because of access. And if you look, even in 2023, at the rate of vaccination or the number of people, frequency of vaccination around the world, there's two things I want you to notice on this slide. One is that Africa still remains largely unvaccinated. And the other thing to notice is that the United States is a lighter shade of green than many of the other countries around it. This means that about 30% of Americans decided they didn't want to take the help from the vaccine. This is a problem of education and misinformation and deciding not to take the help that's available. The problem in Africa is access, and less than 5% of the African continent was vaccinated by the end of 2021. So the great benefit that could have happened in the fall of 2021 was missed. And so if we can't vaccinate the whole world in 6 months, then we will suffer the consequences of repeated waves of variants from whatever new virus is going to be coming through. So in summary, we're in this new era of vaccinology, I think largely characterized by structure-based vaccine design, precision, antigen design, rapid platform manufacturing. We'll use a lot of mRNA vaccines going forward, especially in a pandemic response. There's still a lot of basic research problems to solve. We probably have tools to solve about 80% of our vaccine challenges, or virus challenges, but still need some basic research on immunology and pathogenesis to solve the other 20%. And then there's problems with vaccine access and uptake, and our policies and practices are lagging far behind our technology. And then I want to thank all the academic and government and industry collaborators that helped get this done. By the end of that year of 2020, there was probably at least 1,000 investigators and hundreds of thousands of volunteers who contributed to get this work done. But I want to point out, especially those pictures on the lower left, not just the collaborators at the Vaccine Research Center, but Jason McClellan, who helped with all the structural biology, Kizmikia Corbett, who's now an assistant professor at Harvard with her own lab, helped with much of the animal work and immunology, and those 4 students that I picture there did almost all the bench work needed to start that first phase 1 trial. So the first 10 weeks of 2020 were largely, should be attributed to those 4 young people. And I also want to point out, since we're here together, that we also work with Medigen, a company in Taiwan, and to help them make their first domestic approved vaccine for human use, which was a protein-based vaccine using that spike structure that I showed. So I'll stop there, and thank you very much.
Okay.